Hello, everyone, and welcome to today's session. So let's go through the slides. Yeah. Today's session is about histochemical methods, principles, and application. The first thing we need to talk about, according to the question, it will be histochemistry. Guys, histochemistry is basically based on specific reactions and it's giving or producing insoluble color for light microscope and dense electron dense microscope. Okay, now it's used for two different reasons, either for expression of enzymatic activity in tissue, or is used for detection of various chemical substances. Now, for enzymatic activity, the only one you need to remember is for detection of alkaline phosphatase, which the azodie coupling method is used. We'll see a good picture of it later on the slides, and that's the one I want you guys to memorize it, and the steps will be explained to you, I think, the last slides. Now, for detection of various chemical substances, guys, if you just know these five, you know most of it already. For detection of polysaccharides, there will be best carmine, Alchian blue, and pass reaction, with which pass reaction is like the most important one. For lipids, we will have Sudan dyes, oil red, and for its fixation, if you guys remember from, I think, the first topic for histology, fixation for lipids will be Baker's fluid, and embedding for it will be gelatin, or some professors prefer celluloid, but gelatin still is fine. Now, for phospholipids. For phospholipids, guys, phospholipids can be found in various places of our body, but they are found in myelin sheath as well, and it's a very tricky question which comes in the MCQs. What do myelin sheath possess, and how? They have phospholipids. Phospholipids or myelin sheath, which are inside the myelin sheath, they get stained with fast luxo blue. Now, for DNA detection, we use Fulgens reaction. The Fulgens reaction is very similar to how pass reaction works. Again, the aldehyde group, after the DNA gets exposed and the HCl digestion takes place, then the aldehyde group reacts with the Schiff reagent. Schiff reagent will cause the purple or magenta color that we will see, and that's how we can determine that this was a DNA and determine the sex chromosomes. Okay? Now, for RNA, it will be mostly toluidine blue and methylene blue. Know both of them. It's very useful for you to remember them. Okay? And that's it. That's most of what you need to answer for this question. Next thing, I'm going to remind you guys of these five names, let's say. These five going to be repeated many times during the year. So glycogen, these granules are in the cytoplasm. There will be glycoproteins. That means there is abundant bunch of protein with some glycogen in there. They usually can be found in glycocalyx. Glycocalyx, guys, are the proteins which are around the outer surface of cells, and they're quite important and act as sort of a differentiation between different cells. And there will be glycolipids, mostly lipids, some glycogen in it as well, and they also can be found in glycocalyx. Protoglycans, there is a lot of glycans, proteins in it as well, but mostly glycans. And for GAG, all you need to know, GAG is another name for mucous polysaccharides, and it's found on the cellular excretions. The only reason, guys, I added this is because these are the five different observations you can find for polysaccharides, and you need to know what they mean from now on. If you want to understand what in lectures and in classes the professors are talking about. Now, methods. We already talked about the methods we use in histochemistry, but I'm going to explain four of them a bit more in details, just so you guys will understand it fully. For pass reaction, guys, again, Schiff reagent will get reacted with the aldehyde group, and aldehyde group reaction with this chief reagent will cause a purple or magenta color. So we will see, for example, here is a slide of gallbladder. Pus has been used. We can find reticular fibers here and the mucus here, which are the most important ones, and the basement membrane. Now, past reaction, as we mentioned, it's used for detection of polysaccharides like reticular fibers here, like mucus, like glycogen, and like basal lamins, which we find it in the basal membrane. Okay? Alcian blue, guys, Alcian blue, mostly when we talk about Alcian blue, we're either going to stain mucus or stain goblet cells, which has mucus inside it. But we use it for heparin granules too. And best carmine, as we mentioned, is for staining of polysaccharides, but most importantly for staining of glycogen. Now, when we are staining different slides, sometimes it's very important for us to understand whether that polysaccharides was truly a glycogen or it was other polysaccharides in that sample. And that's when it's important for us to have a test. We have a pass with amylase test, which I'm going to explain to you, and it will differentiate between the polysaccharide that was used, either it was glycogen or not. Okay. How do we do it? We have two different samples. We gather two different container, having the same tissue and same slide. We put it in that container. In one container, we pour water. In another one, we pour diastase. Diastase, guys, is basically an enzyme that will digest glycogen. So if there was glycogen, before we add the pass to both of the container, the glycogen has already been dissolved. So the result will be what? The result will be the water one, the container with water will become purple, detecting that there was, after pass given, by the way, detecting that there was some sort of polysaccharides there. But the diastase one is clear. So that means that the glycogen has already been dissolved and digested. So we will understand that this was a glycogen, the polysaccharide here. But if the water container is purple and the diastase is also purple, that means that the polysaccharides used was not glycogen. Now, if the water container is clear and the diastase cleaner is clear, there was no polysaccharide at all. And there is a misleading sort of statement that comes, it came in our MCQs, I think, in the midterm. And it was saying, what happens if the container becomes clear, the container with water becomes clear, and the container with diastase become purple? That is kind of impossible, guys. So even if you want to memorize these four statements, please do. They're going to be really useful for your midterm and for your questions or quizzes that the professor take in your class from you. Now, slides. T4 slide about gallbladder. I already showed a very small fraction of this picture for you guys, but this is a bigger version, a bit more zoomed out. You can see past reaction has been used here, so we can find the mucus, reticular fibers, and we can see the basement membrane. Now, if we give Alcium blue 2 to detect mucus and gallbladders, you will find something like this. But for detection of the slide T4, which is large intestine, this is one of the most easiest slide, this one and the small intestine. If you guys pay attention to this one specific difference between the two, and you will always be able to detect them. What's the difference? In the large intestine, there will be something like crypts. Crypts will go deep inside, and they have a lot of goblet cells, and therefore mucus inside it. So when you give Alcian blue, they become blue. Now, the difference is visible when you know the difference between crypts and villi. Villi, it's some sort of a protrusion which from the epithelium, it goes in the lumen, and it will be like a finger-like protrusion out of the epithelium to the lumen. And therefore, if we do a transverse cut for large intestine, you will see something like this. You will see sort of a like flower-shaped thing surrounded by remaining parts of the crypts, and then there is connective tissue between them, so they are connected to each other. But if you see cross-section or transverse cross-section of small intestine, guys, you will see the same thing. Difference is between these flower-like structures, there will be white, meaning that there's nothing connecting them. So it's small intestine. Pay very close attention to it. This is like a free question if they give you this in the final exam. They gave to me in the final exam, and it was one of the best things I could ever ask for. This picture you guys can see here, it's a picture that Professor Adriana likes a lot. And it's showing like goblet cells. Here's the goblet cell, ciliated cells, basal cell, and stem cell. And the two here as well. Forgot the name, but these two as well. This picture is very important. Know how to draw it, even if you're not her professor, because it's possible in the final exam she became your examiner, and she likes to ask this in the final exam, as well as in class. But yeah, so know how to draw this picture, guys. Now, for histochemistry of lipids, guys, I already talked about a bit of it in the first slide or second, but I want to dive into it a bit more. You guys will observe lipids as either fatty acid, cholesterol, neutral lipids, or most importantly, phospholipids. But the methods to stain lipids will be Sudanese, oil red, Luxo blue, and Nil blue. As you can see here, we have a oil red. This is a good slide of it. And here it's part of the brain, and it has been stained with fast Luxo blue, especially for staining white matter and myelin sheet. As you guys can see, if you zoom in here a bit, you will see there is a red nuclei. So that's very important. Enzyme histochemistry. I put this slide, it's a bit more details of what we already talked about of azocoupling. You just need to remember that the catalytic histochemistry, it will determine the location of the enzyme activity by staining the substrate that is being catalyzed by the enzyme. And you need to remember that every time we want to use enzyme histochemistry, we are trying to detect alkaline phosphatase, especially, for example, in PCT, proximal convoluted tubules in kidneys. It's very important because they have a lot of alkaline phosphatase, and we will just give an azodicoupling and stain it. Speaking of kidneys, this picture you guys see here is a kidney. As you see, it's very difficult to determine that this is a kidney. However, because of color, we cannot determine that this is an azodicoupling. But this is a misleading question because some professor will ask, it's a kidney of which animal? Is it a human or is it another animal? The answer to this question, it will be, it's a kidney of a mice, literally. And you guys need to remember also that it's always this process, this method is done when it's fast red used by diazonium salts and the buffer needs to be pH of 9. Okay? And this is what I just said in the other slide as well, just a bit more organized, showing the process step by step of how azodecoupling works. But again, same thing I mentioned in the last slide, it just seems more organized, so I put it there for you guys. And that's it. That's how you're going to answer this question if you get it in the final. And I wish you guys the best of luck. Hearing and end the slideshow, and we will end the recording.